All right, let's talk about one of the most important topics in biology, enzymes. If you can master this, you're building a huge foundation for acing your exams.
So grab your notes, get focused, because in the next few minutes, I'm going to walk you through everything you absolutely have to know about enzymes to smash your biology GCSE. We're cutting out the fluff and getting straight to the essential exam-winning facts.
So here's our game plan. We'll kick things off with what an enzyme actually is, then we'll meet the key players in digestion. After that, we'll get into the exact conditions they need to work, cover the 4 food tests you have to know, see how all those good nutrients get into our blood, and we'll finish up by building the perfect exam answer. You ready? Let's do this.
Okay, first things first. Part 1. What on earth is an enzyme? Before we can talk about digestion or anything fancy, we have got to nail these basics down.
The single most important definition you need to remember is this. An enzyme is a biological catalyst. Biological because it's made of protein, and catalyst because its entire job is to speed up chemical reactions, like digestion, without getting used up itself.
So how do they actually work? Well, we use something called the lock and key model. I want you to think of the enzyme as a lock, and it has this very specifically shaped part called the active site. The molecule it works on, which we call the substrate, is the key. And just like a real lock and key, only the right key, the specific substrate, is going to fit that lock. That's why they're so specific. It's why amylase only breaks down starch and won't touch protein.
But what happens if you damage that lock? Well, that's called denaturation. If the temperature gets way too high or the pH is too extreme, that delicate protein structure just falls apart, and the active site changes shape for good. The key doesn't fit anymore, and the enzyme is totally useless. This is an absolutely critical idea.
Right. Now that we know the rules of the game, let's meet the key players in our digestive system. There are 3 main families you have got to memorize for your exam.
First up, the carbohydrates. No prizes for guessing what they do. They break down carbohydrates. A really key example is amylase, which takes a giant starch molecule and chops it into a smaller sugar called maltose. Then, another enzyme called maltase comes along and finishes the job, turning that maltose into glucose. And that's the simple sugar our cells can actually use for energy.
Next, we have the proteases, and they're the ones that break down proteins. You've probably heard of pepsin. It loves the super acidic conditions in your stomach, where it gets to work breaking down big proteins into amino acids. Later on, in the small intestine, another one called trypsin helps to finish off the job.
And our final family is the lipases. These enzymes have one crucial job, to break down lipids, that's just a fancy word for fats and oils, into 2 smaller bits, fatty acids and glycerol. It's as simple as that.
Okay, so we've met the enzymes, but for them to actually do their job, the conditions have to be just right. This whole section is about how our body creates that perfect environment for them to work their magic.
This brings us to a classic exam question. Why can't an enzyme like pepsin, which works perfectly fine in the stomach, also work in the small intestine? Just take a second and think about it. The answer is all about the conditions.
And here's why. It's all about pH. I mean, look at this difference. The stomach is incredibly acidic. We're talking pH 1 or 2, which is the perfect condition for pepsin. But the small intestine is alkaline, around pH 7 or 8. If pepsin were to move in there, that massive change in pH would cause its active site to denature, and poof, it would stop working completely. Enzymes are total specialists, perfectly tuned to where they live.
So how does the body make that pH change so quickly? Well, it uses a crucial helper molecule. And I need you to listen carefully here, because this is a key point. Bile is not an enzyme. It's such a common mistake people make. Bile is made in the liver, stored in the gallbladder, and its job is to help out the enzymes in the small intestine.
Bile has 2 absolutely vital jobs. First, it's an alkali, so it neutralizes all that stomach acid as food enters the small intestine. This creates the perfect alkaline pH for enzymes like trypsin and lipase to get to work. And second, it emulsifies fats. Now, all that means is it breaks down massive blobs of fat into tiny little droplets. This gives the lipase a much bigger surface area to attack, and it massively speeds up fat digestion.
Okay, this leads us nicely into the practical side of biology, the 4 key food tests. This is the real detective work, where you get to prove which big food molecules are actually in a sample.
First up, the test for starch. This one is so simple. You just add a few drops of iodine solution to your food. If there's starch in there, the orangey-brown iodine will turn a deep blue-black color. No starch, it just stays brown. Easy.
Next, how do we test for sugars? For this, you add some Blue Benedict's solution, and then you have to heat the sample gently in a water bath. If there's sugar, you'll see a color change. A little bit of sugar will turn it green or yellow, but a lot of sugar will turn it a deep brick red color.
And what about protein? For that, we use the Biuret test. You just add Biuret solution to your sample. If the solution goes from its normal blue color to a definite purple or lilac, you've got protein. It really is that straightforward.
And finally, the test for lipids. Fats. For this one, you first add ethanol and give it a good shake. This dissolves any fat. Then you pour that liquid into some water. Because fat and water don't mix, if there are any lipids present, they'll form a cloudy white layer. We call that an emulsion.
Okay, so all our big food molecules have now been chemically chopped up into small, soluble molecules like glucose and amino acids. But how do we get them out of the intestine and into our bloodstream so our body can actually use them? Well, that process is called absorption.
The inside of your small intestine is lined with millions of these tiny finger-like things called villi, and they are total superstars when it comes to absorption. Their shape gives them a huge surface area, which is made even bigger by microvilli on their surface. Their walls are super thin, just one cell thick, for a short diffusion path. And they have a fantastic blood supply to whisk those nutrients away, which keeps the concentration gradient steep.
So after all of that, how do you pull it all together for the exam? When you get that big question on digestion, what does a perfect top mark answer actually look like? Well, here is your blueprint.
These are the killer points you have to hit. First, state that enzymes are specific biological catalysts. Second, explain that digestion breaks large, insoluble molecules into small, soluble ones, and give an example, like protein being broken down into amino acids. Third, and this is crucial, you have to mention the two roles of bile, neutralizing acid for the perfect pH and emulsifying fats. And finally, listing those 4 food tests shows the examiner you really know your stuff. If you can nail these points, you are absolutely golden. It's pretty amazing when you think about it, right? All these tiny little protein machines are the only reason we can get any energy at all from the food we eat.